All right, hi you guys. Welcome to our second lecture. This one's on DNA replication. And so this is when you would take all of your chromosomes and copy them, basically, right? So a cell does need to replicate itself in order to ensure that it survives for multiple generations, right? This is true for bacteria. This is true for humans, right? So for instance, like the survival of the larger organism, like the human itself, or the species, like human race, or even the individual cells. So, like, if a liver cell or a heart cell or a brain cell, right? Um, so, in order to preserve the generational survival of these cells, they need to take part in DNA replication. So, it's sort of like if you know you're going to die, you need to copy the parts of yourself that will ensure your survival in the next generation, and that would be your DNA. And so let's talk about when to replicate. When would you actually want to replicate? And so there's really two reasons, right? A cell should replicate itself when some adjacent cells around it become damaged. So, like, let's say you cut your skin or something, right? Those skin cells are going to send out signals that they've been damaged, and that's gonna initiate replication and cell division in the cells around the damaged cells. Or, right, so that's one if there's damage done, or if the organism is growing or developing or something, right? So babies are a great example, right? Their cells are replicating themselves all the time to grow and develop to become bigger and stronger. And so what you see when this happens, when you see development, growth, development, and cell replication, um, what we see is a direct duplication of all of the contents in the cell. So it's not just DNA, right? So it's either the mitochondria are going to duplicate, the um, the chloroplast if it's in a plant cell, right? The lysosomes, the vacuoles, all of that stuff is going to replicate, right? But the important part, the most important part, is the DNA is going to replicate itself, right? So you get a direct duplication of all the stuff in the cell, the DNA that's in the cell. And then if it's a cell like a heart cell or a liver cell or a lung cell, right, the job specificity of that cell will also be replicated and shown forth in the new cell that's made. The type of replication process it goes through. So the it would be through mitosis. And so mitosis is making identical copies of the same cell. That's a way to think about it. Okay. When the organism needs to reproduce, so this is the second reason that a cell would need to replicate, is when it needs to reproduce, right? And so this would be an example of multicellular organisms when they need to reproduce, things like eukaryotes, right? So when bacteria need to reproduce, they literally go through like a stage like mitosis. They just duplicate themselves and everything inside themselves. But when organisms like eukaryotes, especially ones that have male and female organisms of the same species, that's when you would need to replicate your cell as well. And so what ends up happening when you need to reproduce and you need to make offspring that are unique and different from the parents, right? Is that you would duplicate the DNA, but then you would swap alleles between the chromosomes that you got from mom and the ones you got from dad. So consider this, right? Go back to our last slides, right? Our last lecture. And we saw that the left shoe we got from dad, and then the right shoe for all the pairs of chromosomes we got from mom. Um, well, each one of those shoes is going to have a different set of alleles, even though the homologous chromosomes, right? The pair of Dockers is going to have the same order and sequence of genes on it. Each one is going to have its unique set of alleles, right? And so what might end up happening when the organism goes to reproduce is that it will duplicate all of the DNA, but then it swaps alleles. It'll say, okay, I'll take some alleles from mom's chromosomes and put them on dad's, and take the ones from dad and put them on mom's. And so it ends up creating this enormous amount of diversity in just one generation. Okay. And so essentially, what you are, if you think about it, like half of you is like half of the set of chromosomes that you got, right? So let's say that the set of chromosomes you got from your dad, right, is actually an amalgamation. It's a mix of the chromosomes that he got, like the alleles that he got from his mom and the alleles that he got from his dad. So one set of your chromosomes is actually a mix of your two grandparents. The other set of chromosomes that you have is a mix of your other two grandparents. And it's kind of crazy how that works. Okay. So anyways, swapping of alleles between chromosomes, and then it goes through meiosis. And meiosis is cell division that results in haploid sperm and egg. And so let's talk about meiosis, right? Meiosis is only for sexual reproduction. Meiosis is only for sexual reproduction. And so you have a duplication of the DNA. So if we're counting this down, right? Let's count out the chromosomes. So you have, before any of this takes place, you have 2 sets of homologous chromosomes. One set you got from dad, one set you got from mom. Those homologous chromosomes are going to go through DNA replication. Boom. Now you have 4 sets of chromosomes. The 2 you got from dad and the 2 you got from mom, they're going to switch alleles, but there's still 4 chromosomes. Then it's going to go through cell division twice. So you're going to end up removing half and then half again. And so if you think about it, right, you're whittling it down just to 1 set of chromosomes if you divide it twice, right? So here you have 4 chromosomes after DNA replication, right? It goes through cell division, right? So now each cell only has 2 sets, and then those cells go through cell division again, and now each cell is only going to have 1 set of homologous chromosomes. And what that does is it makes 4 unique haploid sperm or eggs, right? And so each sperm or each egg only has 1 set of chromosomes. And so when the other one comes in, that's what makes an entire human being. Okay, either way, the cell is going to have to replicate its DNA. So let's talk about the models of replication. So before people actually figured out how DNA replicates, they came up with several models for it. And I'm going to give you some analogies for these. So the first model is a conservative model. Okay, and I use peanut butter and jelly sandwich for this, right? Because DNA has 2 halves, right? DNA has 2 strands. Okay, it's got 2 strands. So we can think about those strands kind of being complementary to each other, like peanut butter and jelly in a sandwich, right? So when we go to replicate in the conservative model, we take a peanut butter and jelly sandwich, we open it up and look at it, and we say, well, look, this piece of bread has peanut butter on it, so I'm going to make a new piece of bread with peanut butter on it. Then you look and you say, oh, look, this piece of bread has jelly on it. I'm going to make a new piece of bread with jelly on it. And then, boom, you put the new peanut butter and new jelly half together, and you have a brand new peanut butter and jelly sandwich that's an exact replica of the other one, but one is old. The entire molecule is old. The entire double-stranded molecule is old, and the other one is brand new. So the other double-stranded molecule is brand new. Okay, so that's taking a peanut butter and jelly sandwich, doing that. Now the semi-conservative model, right? Spoiler alert, is actually how it works. So the way the semi-conservative model works is you have a peanut butter and jelly sandwich, right? The DNA halves separate from each other. You look at the peanut butter half and you say, okay, that needs a jelly half. So you make a new jelly half and put it on top of the old peanut butter half. You then look at the old jelly half and you say, man, that needs some peanut butter. So you take a new piece of bread, put peanut butter on it, and then put that on the old jelly half. And so what you end up with when replication is done is you end up getting 2 sandwiches, half new, half old, right? One half is new peanut butter on old jelly. The other sandwich is old peanut butter on new jelly. Hopefully I said that right. So anyways, hopefully that makes sense. And then the dispersive model was another one proposed by some I don't know, brainless idiot that just put it. So dispersive model is like this. Peanut butter and jelly sandwich, open it up. You're going to scrape some peanut butter off one half, put that on a new piece of bread, and then add some more new peanut butter to help fill it out. Then, nobody would ever do that. But anyways, we can draw this out like in an artistic depiction here. So conservative model, right? Peanut butter and jelly sandwich. Let me get my little swishy pointer. So peanut butter and jelly sandwich, right? We're going to open up one and make a brand new copy from it, right? Just by looking at it. So one will be new and then one will be old. Okay. Then the new one's going to go replicate. It'll make 2 just like itself. The old one will then replicate and it will continue to exist, but the replicate that it makes will also be a new one. All right. Really, I just want you guys to focus on the second column, right? Okay, so the semi-conservative model is you have a DNA molecule, double-stranded, right? It's going to open up, and the peanut butter half, right, the dark blue half, is going to get a new jelly half. And then the other side, the old jelly half, is going to get a new peanut butter half made. And so what you end up with is 2 molecules, half new, half old. Okay? And then dispersive models, like you scrape some off of one, put it on another, and then, that just doesn't look uniform. That doesn't look nice. That looks like a lot of work, a lot of extra work. Okay, moving on. So how this works in prokaryotes and eukaryotes. So no matter what, DNA is double-stranded, and DNA needs an origin of replication. All right, so in prokaryotes, which have circular chromosomes, there's only going to be one origin of replication, and that's where the two strands of DNA. Remember how I said sometimes they'll draw the two strands of DNA really simply, right? If they're just talking about replicating the entire strand. So they drew each strand as a blue circle. And so at the origin of replication, the two strands are going to separate, right? And then the new strands that are being synthesized are going to be this lighter color of blue. And then as you can see, you got two replication forks, right? Those are shown by the red arrow. A replication bubble is just the bubble in the strands that is forming from making a new DNA strand. And then the replication forks move towards each other until they meet in the middle, and then pop, you get two new chromosomes, right? Half new, half old for each of them, right? And here's an image of that taking place, an electron microscope image of that taking place. Notice the two red arrows here show the replication fork, right? That's where the two strands are being separated actively. And so here's the artistic depiction of it. And then here is the actual electron microscope image. Okay, but with eukaryotes actually, because their chromosomes are so long, right, they actually have multiple origins of replication. And when you take your, if you take your genetics class, right, you'll go into extreme detail on there's specific nucleotide sequences that attract the enzymes to these locations that start to open up the two strands from each other. I'm not terribly interested that you know that, but I think it's interesting, but it's not necessary to understand the rest of this. So we've got multiple origins of replication in eukaryotes, right? Those replication bubbles will then start to move towards each other. So as the replication forks from one origin of replication move towards the replication fork of the other origin of replication, eventually they will meet in the middle and you'll end up with two daughter DNA molecules or DNA strands, half new, half old. All right, so let's just talk about how this whole thing gets going. Okay. All right. So there's a nucleotide sequence specific start site. So that just means the order and sequence of nucleotides on that DNA strand are going to attract all of the molecules and the machinery that helps with replication, right? Okay. Helicase is going to separate the two strands. Okay, so helicase is an enzyme that's going to move at the replication fork that helps to separate the two strands. Okay, single-stranded binding proteins will bind to the strands and keep them from zipping back or gluing together. Okay, helicase is working to separate the two strands, while topoisomerase is moving ahead of the replication fork. It cuts the DNA and relieves the tension and then glues the DNA back together after the tension has been released. And I'm going to show you an example of that with a pair of shoes up here in my video. Okay, so hold on, I'm going to pause it and grab my shoes. Okay, I've got my shoes. That was fast, right? I got my shoes, right? And so you'll notice that shoestrings have two strands, right? And that they're twisted together, right? So I just spun it a little bit, right? So as you can see, the shoe has its DNA strands up at the top, and they're twisted together in a helix, in a double helix, right? So you can imagine, and I'm going to try to rest the shoe on top of something like my water bottle. So I can, so here we go. All right, so here we go. Keep saying here we go. So as the origin of replication opens up the strands, right, and separates them from each other, down here, right, the shoestrings, the loops are getting closer and closer together. It's creating a lot of torque down here, right, as the strands separate. Look, it's even spinning my shoe the other direction. So a lot of torque is created from separating the two strands together. It kind of like twists the portion that's not separating. It twists them closer together. The turns get closer together. There gets to be a lot of tension. And so topoisomerase is the enzyme that moves ahead of the replication fork. It cuts the DNA, literally cuts it, and allows it to spin. And then once that tension is relieved, it glues the two strands back together. Okay, so it's kind of a cool enzyme. And then you've got RNA primase, right? RNA primase comes along, and RNA primase makes an RNA primer on the template DNA strand. So the strand that's about to be replicated, RNA is coming along and laying down a primer on that. Specifically, right, so when RNA primase is laying down the RNA primer on the template strand, what we mean by that is the template strand is going to be both sides, right? So the template DNA is that's going to be read and copied, and there's two sides, right? Because there's two strands. So there's going to be a leading side and there's going to be a lagging side, and you'll see this when we like actually get into the diagrams of it. All right, so here's the first diagram. Okay, this is the origin of replication. So we haven't even brought in the machinery yet to replicate the DNA. directly. This is just the stuff to get it started. All right, so notice you've got the 2 strands are separated. The origin of replication is going to be over here by this 3' end of the top strand, okay, and also by this 5' end of the bottom strand. Okay. The 2 strands were separated by helicase, right? Helicase is coming along and unzipping it. As helicase moves forward, these strands, which in this image are not shown as twisted, are getting more and more torque placed on them. Topoisomerase just cuts the DNA strands, allows them to unwind, and then glues them back together. So it moves. See, you can see the little cut there, right, by the topoisomerase, right? So it cuts and glues. And then you've got these single-stranded binding proteins that are binding to the 2 strands that keep them from zipping back together because they want to, right? The stable state for these 2 strands is together. That's why DNA is so stable, is because when it's double-stranded, it can sit there for decades and not degrade. But the 2 nitrogenous bases have to be glued together. So anyways, they want to glue together, right? There's a magnetic attraction between the 2 strands. So these single-stranded binding proteins keep those from happening, from gluing back together. All right, let's talk about the enzyme that's going to do it all here. So DNA polymerase 3, there's several DNA polymerases, as you probably can derive, but there's the 3rd one is the most important. It's probably named 3 because it was the 3rd one that was discovered, to be honest. Okay, so DNA polymerase 3 requires this RNA primer to be laid down. So RNA primer is going to lay down some primer here, and then it's also going to lay down some primer on this strand. But the DNA polymerase 3 needs that primer in order to start polymerization. Okay. It also is going to require DNTPs, and you're like, what is a DNTP? Well, let's figure it out, right? D stands for deoxyribose, N stands for nucleotide, T stands for tri, and then P stands for phosphate. So if you think about it, a DNTP is literally just a single nucleotide with 3 phosphates bound to it, right? So you're going to have ATP. Look at that. So you're going to have adenosine triphosphate, or deoxyribose adenosine triphosphate, deoxyribose guanine triphosphate, deoxyribose cytosine triphosphate, and deoxyribose thymine triphosphate. And that's what those are. So these are individual monomers with a great deal of free energy attached to them. So these are the guys that actually power the polymerase to do its job. They power the polymerase to do its job and make a polymer of DNA. Because think, ATP has a ton of free energy. So does GTP and CTP and GTP. They all have a lot of free energy. So the polymerase is going to use that free energy to do its job. And then also, these are the building blocks of this new strand of DNA that it's making. Okay. So this process is going to be a dehydration because it's got to dehydrate it to make a polymer, right? It's going to be endergonic because it's going to require energy from outside to come in. And it's anabolic because you're building a new DNA strand. The only thing that it really needs is it needs a template DNA strand. So it's going to read one thing, and then it's going to put the complementary base pair as the next thing. And I'll show you that in a different image here. Okay. So the important thing is that it has to follow this rule, though, and this is the rule of forming DNA polymers, is that new nucleotides can only be added onto the 3' end of the previous nucleotide. So go back to our RNA polymerase, right? Notice there's a 5' end of the RNA primer, and then there's a 3' end to the RNA primer. That means DNA polymerase 3 can only put new nucleotides onto this 3' end. That means it can only add nucleotides going in this direction towards the replication fork, right? And when we get to this bottom strand, it's going to have a different set of rules because the bottom strand is facing the opposite direction. So that one's going to be the tricky one. That's the lagging strand. The leading strand, DNA polymerase 3 can just come in, bind to this 3' end, and start stacking nucleotides in a 5' to 3' direction. Okay, so new DNA can only be synthesized in a 5' to 3' direction. New nucleotides can only be added onto the 3' end of the nucleotide before it. And there's some organic chemistry involved with why that's the fact. Okay, fun fact though, humans copy about 50 nucleotides per second. Bacteria can copy up to 500. So which one do you think makes more mistakes? Bacteria, right? So that's how bacteria have actually been able to stick around and evolve over the millennia, over the millions of years, is that bacteria on purpose have DNA polymerases that are a little less accurate, and so they create mutations and create variants in bacteria, even though bacteria don't have sex and they don't reproduce like that. They just make direct copies of themselves. Okay. I digress. All right, let's move on. So this is an image showing you how these DNTPs, right? The DNTPs come in and power the DNA polymerase. So what we have here is we have a template strand, right? Notice the template strand is running 3' to 5'. So the dark blue is the pre-existing strand, or if you will, it's the old peanut butter half of the DNA molecule. And then the DNA polymerase is coming in and it's creating a new jelly half by looking at the peanut butter half. So it sees a T, it sees a T, then that sounds funny. It witnesses a T, right? It reads a T, and then it makes or brings in an A that's complementary and pairs it to that T. So that's making the jelly side to the peanut butter. So new jelly nucleotide to an old peanut butter nucleotide. And then it reads a G and it places a C down, right? And so it's synthesizing this new strand in a 5' to 3' direction. So the new nucleotide that's coming in here, right? This new DNTP, right? This one happens to be a TTP, right? Or a deoxythymine triphosphate. So it's got 3 phosphates. So it's going to come in and power the reaction. Notice that the phosphate is bound to the 5' end of the nucleotide. And that phosphate is going to go attach itself to the 3' end of the existing jelly nucleotide ahead of it, right? And so the DNA polymerase, this is step 1. Step 2, boop, DNA polymerase uses those phosphate groups to power this reaction. And then a pyrophosphate or 2 inorganic phosphates pop off and they'll go be recycled. But notice here, right, that thymine group, that thymine triphosphate, got attached to the 3' end of the one before it. Boom, clicks in, right? And then the next one would come in. Maybe you guys can tell me what would be the next nucleotide that would fit here. Guanine triphosphate. Yep, guanine triphosphate would come in, and its phosphate group would be bound to the 3' end of the nucleotide ahead of it, and then it just goes on like that. Okay, so what we're having, just an overview here, right, is what we've looked at so far up to this point is we've looked at the origin of replication, how the 2 strands are separating. We know that DNA polymerase 3 is going to bind to the RNA primer, and it's going to just start adding nucleotides in a 5' to 3' direction, putting new nucleotides onto the 3' end of the nucleotide before it. Okay, that's great, right? But what we also have to look at is what's happening on the lagging strand. Replication really tricky. It's abstract and it's tricky to wrap our minds around, right? But this origin of replication, right, is going in 2 directions, right? You've got one direction going to the left, one direction going to the right. And notice, right? Notice that the leading strand going to the right is opposite to the leading strand going to the left. And you can see why if you look down at the directionality of the template strand. Up on the top here, right, the template strand is running in a 3' to 5' direction, which means the new strand that's anti-parallel will be created in a 5' to 3' direction continuously. So there's no problem reading 3' to 5' and copying 5' to 3'. Going the opposite direction, right, the 3' end of this leading strand is on the bottom, right? And the 5' end is on the top. And so the DNA polymerase will be able to bind to the 3' end on the template strand, read 3' to 5' while it's creating a new copy 5' to 3' anti-parallel. And so now we have to deal with the lagging strand. What's going on with the lagging strand? Okay, so let's we'll get there in a second. We'll take a deeper look at what's happening on the leading strand. So here we have the RNA primer was laid down by RNA primase, right, in a 5' to 3' direction, right? And then DNA polymerase 3, which is shown by this little orange C-shaped character here, it's binding to the end of the RNA primer, and it's adding new nucleotides in a 5' to 3' direction. It's attaching the new nucleotide onto the 3' end of the previous nucleotide. Okay. Helicase, single-stranded binding proteins, they're all doing their job, right? And so on this leading strand, helicase is unzipping it, and the DNA polymerase 3 is just going to be continuously elongating in a 5' to 3' direction. Okay. Now let's deal with the lagging strand. So now we're going to look down at the lagging strand. So here's the lagging strand, right? So if you can look here, right? we're gonna be looking at the bottom strand, right? The top strand, no problem. We can continuously make nucleotides 3' to 5'. But down here on the bottom strand, right, we have this problem, and the problem is that the origin of replication is moving to the left. It's moving that direction, right? And the problem is that the strand that's being read, the template strand here, right, can only be read in a 3' to 5' direction because it needs to synthesize new nucleotides 5' to 3'. Remember, that's the rule that it has to follow. It can only add new nucleotides onto the 3' end of the one before it. And so RNA primase is going to come along and it's going to lay down a primer, and then the DNA polymerase is going to come in and bind to the 3' end of that primer and start adding nucleotides going towards the origin of replication, away from the replication fork. And so what that means is it's going to run into the primer for the leading strand going the other way towards the other replication fork. It's going to run into that leading strand, and then it's going to hop off and go find the RNA primase again. So here, that first strand, right, RNA primer is laying it down. DNA polymerase is going to come in and bind onto the 3' end of that RNA primer, and it's going to add nucleotides in a 5' to 3' direction until it hits that primer for the other origin of replication. Okay, what it's doing as it's making this chunk right here, right, because this is going to be done in chunks. That chunk is called an Okazaki fragment, and the Okazaki fragment is named after a husband and wife research team in Japan that discovered these fragments. Okay. So once it hits that primer, it's going to detach. It's going to find the next RNA primer closer to the replication fork, and then it's going to copy in a 5' to 3' direction until it hits the previous primer. Boom. And then it's going to hop off and go ahead again, and it's going to keep doing it. So it looks like this. It copies, picks up, lands ahead towards the replication fork, copies, bumps into the primer before it, picks up, moves towards the replication fork. And so the way I see this, like in my head when I took genetics, is I was like, oh, okay, well, the DNA replication is kind of like a skateboard. One foot stays on the board, right, on the leading strand, and it just copies continuously. It just rolls forward. The other foot has to pick up, set down, pull back. Pick up, set down, pull back. Pick up, set down, pull back. And so that's the difference between leading and lagging strands. Now, there's another DNA polymerase that comes in, and its sole purpose is just to take the RNA primer that's there and replace it with DNA nucleotides. That's it. That's its only job. Okay. And then DNA ligase is another enzyme that comes along, and it's going to join the Okazaki fragments together, right? If you think about it, right, we can only polymerize by adding nucleotides onto the 3' end. As soon as you get to the next RNA primer, there's going to be a gap there. DNA polymerase cannot catalyze that gap. It can't close it up. So it's going to hop off and go find the next RNA primer and replace it there, like it's doing in this picture. And then once it's done its job, DNA ligase comes in and it joins those gaps. It seals those gaps. So notice this gap up here, right, is sealed already by that DNA ligase. And then the DNA ligase comes up. So DNA ligase is joining the Okazaki fragments together until the lagging strand is complete. And so here's an overview of the DNA replication, right? And I'm going to have this image on an exam. And what I want you guys to do is be able to identify and tell me what's happening at each stage, right? Okay, so if I say, what's happening here? Oh, that's DNA polymerase laying down nucleotides on the leading strand continuously. What's happening over here? That's DNA polymerase on the lagging strand, laying down nucleotides until it hits the RNA primer in front of it. Then it's going to pick up and go to the next primer. So you can see everything happening here, right? Leading strand is being copied continuously, right? The lagging strand has the RNA primase picking up and jumping back and following. The RNA primase is following the origin of replication as it moves, or sorry, the replication fork as it moves away from the origin of replication. So this replication zipper is moving away from the origin of replication. Primase is following it on the lagging strand, laying down a little set of RNA primer, picking up, moving towards the replication fork, laying down a little set of primer, picking up. And then DNA polymerase 3 is just creating Okazaki fragments. It finds the RNA primer, it makes nucleotides until it hits the next RNA primer, picks up, moves towards the replication fork, finds the next RNA primer, adds new nucleotides until it meets the next primer, picks up, and so on and so forth. Then along comes polymerase 1, and it's going to replace all of the RNA primers with DNA, and then ligase is going to seal up those Okazaki fragments. All right, and so after replication is over, right, we can say, well, what happens, right? So in prokaryotes, the DNA chromosome, the circular chromosome, is going to go through replication. You get the chromosomes separate, and then it's going to go through binary fission, where the one cell becomes two cells, each with their set of chromosomes, right? So in a lot of ways, this is like mitosis, right? It's like mitosis, but in prokaryotes. Now, we typically think of mitosis usually when we talk about eukaryotes. All right, so like I said, there's two reasons you go through DNA replication in eukaryotes. One is if you want to do meiosis, right? That's to create gametes like egg and sperm. The other reason you go through DNA replication is if you need to copy your diploid cell and make an identical copy. And so what you have in mitosis, you have each chromosome is replicated. That's where it goes from half an X, right? Like you're seeing here, to a full X, right? So now that's got each side of one of these X's is identical to the other side of the X, right? And so this would be like this would be your homologous chromosome you got from dad that's now been duplicated. And then on the bottom would be the homologous chromosome you got from mom that's been duplicated. Another one you got from dad right here, another chromosome, and it's a homologous counterpart here. And so when notice the chromosomes, right? There's a big red one, a short red one, a short green one, and a big green one. So notice that when we go through our cell division, our mitosis, each new cell, right, once you separate those identical copies from each other, each new cell is going to end up an identical copy of the other one that looks just like the parent, right? So each one's going to have a long red one, a short red one, a short green one, and a long green one, right? Now go over to meiosis. Meiosis, remember I said they're going to exchange, right? They're going to exchange alleles between homologous chromosomes, and they're going to divide twice. So first cell division, right? Now they both have a set of chromosomes. Second cell division, now there's going to be 4. So what you're only following one side of this, right? So that one side divides again, right? So in reality, though, this side is also dividing again. So there'll be 4 gametes, and that'll be either 4 sperm or 4 egg. And in this case, it looks like it's an egg that meets a sperm, and then now you have a zygote and a diploid organism. Okay, and that's it for DNA replication.